guys, we have a big mind map to go through, and our theme here is DNA, why it's great for information storage and information transfer. The first question is, how do we know that DNA has these purposes? We found out that DNA is the genetic material through a few experiments. The first experiment was by Fred Griffiths, and he first identified that there is something, he called it a transforming factor, that can take bacteria and change their phenotype. He observed this when he took some deadly bacteria and killed it, took some non-deadly bacteria and let it live. He put the two together, and it was like one of them taught the other one how to be deadly. He injected a mouse with this mixture, and the mouse actually died. So somehow, one bacteria transformed the other. We know that that transforming factor was DNA because of the work of Avery, McCarty, and McLeod. They did the same experiment but added an enzyme that destroyed DNA and found the transformation was stopped. This rough bacteria kept its non-deadly phenotype. That means that the transforming factor must have been DNA itself. We know that DNA has these jobs because of those two experiments. What we're going to learn about next is DNA's structure. We discovered its structure through the work of Watson and Crick, and they took some of the work of Rosalind Franklin to contribute towards their results. And they found that DNA is an anti-parallel double helix. So here is the overall view of DNA, and here's kind of a chemical zoom in. This is the ladder model. The only thing wrong with it is it's not twisted. We know that DNA is double-stranded, and each strand is made of a sugar phosphate backbone with nitrogen bases that point towards the middle. What elements are in this molecule? PCHON. Important takeaways here are that every phosphate group is negative, so that gives DNA a net negative charge. Another important takeaway is that these backbones are very, very strong because they're held together by strong covalent bonds. Meanwhile, the inward-facing nitrogen bases are held weakly together by hydrogen bonds. Our last takeaway is, what does anti-parallel mean? It means one strand has the 3' end pointing up and the other strand has the 5' end pointing up. How do we learn what to call which end? We label the carbons of our pentose sugar. This is the pentose sugar deoxyribose. There's an oxygen at the top, and then we count clockwise. 1', 2', 3', 4', 5'. This is our 5' end. What we know so far from experiments is that DNA is the genetic material, and it is shaped like an anti-parallel double helix. We're going to use this structure of DNA and the base pairing rules to explain how DNA replicates and how it is used in protein formation. DNA is going to be found in prokaryotes chilling in the cytoplasm. It's going to be circular. DNA in eukaryotes, like plants and animals, is going to be restricted in a nucleus. It's going to be linear, meaning like a straight line that is not connected to itself. And it has proteins called histones. What that looks like is histones exist in the DNA, and DNA wraps around them like a little spool of thread. Here's another histone. Let's color that one in. And DNA gets wrapped around it. DNA replication is semi-conservative. That means if we take old template DNA and replicate it, our new strand will be half the old template and half the brand new strand, whether it be the leading or the lagging strand. We found this out through the work of Messelson and Stahl. They labeled DNA by growing it in heavy nitrogen, and then they put it in a light nitrogen environment. That let them observe how much lighter does DNA get with every round of replication. At first, when they put all this heavy DNA in, it was all heavy. After one round of replication, they found that it was all somewhere in between heavy and light. And after their final round, they found that half the DNA was totally light and half of it was in between. That meant that some of the DNA was new, and some of the DNA was old and new. If we zoom in on a replication bubble, we're going to see how do enzymes use DNA's structure to perform semi-conservative replication. First of all, DNA is anti-parallel. So that means the top and the bottom are running in different directions. That causes the DNA replication process to happen in a leading strand and a lagging strand, because DNA polymerase 3 can only add on to the 3' end of the growing strand. The 3' end of this growing strand conveniently points towards the fork, but the 3' end of this lagging strand actually points away from the fork. In our leading strand, what has happened is first, DNA helicase unzipped these weak hydrogen bonds. Then RNA polymerase added an RNA primer. After that primer was added, DNA polymerase 3 does the work of adding new nucleotides to the 3' end using the complementary base pairing rules. On the bottom, the same process has happened, but moving in the opposite direction. So first, primase adds RNA primers, then DNA polymerase 3 adds DNA onto those primers towards the 3' end. This forms separate segments called Okazaki fragments that will have to later be welded together by the enzyme ligase. The final step is proofreading, and it's when we take out the RNA primers and fix any mutations. That's the job of DNA polymerase 1. The parts of DNA structure that were convenient for us to use were, number 1, hydrogen bonds are weak and easily breakable by helicase. Number 2, DNA is anti-parallel, and that means that one strand will be leading towards the fork and the other will lead away from the fork, and that is our lagging strand. And the last part of DNA structure that we used is the complementary base pairing rules. When DNA polymerase 3 adds new nucleotides on, it always uses these base pairing rules. One more fun fact here is the role of telomeres in this process. So the concept we want to focus on is that DNA protects itself. It has these little end caps called telomeres, and they stop replication from degrading chromosomes. If telomeres didn't exist, every single time a chromosome replicated, some of it would basically fall off of the end and genes would be lost. So it's a good thing we have these telomere end caps on. We are going to move on to the other function of DNA. So we are going to do the 3-step process of building a protein from DNA in a eukaryotic organism. Let's go to step 1. It's transcription, and it's when we, in the nucleus, transform DNA into messenger RNA. This is in the nucleus only, remember, in eukaryotic cells. This looks a lot like DNA replication because it is. In transcription, a bubble forms, and an enzyme called RNA polymerase adds complementary nucleotides. It knows where to go because a promoter region containing a Tata box bound to a transcription factor. This is just a regulatory protein. And RNA polymerase adds new nucleotides onto the growing 3' end of the messenger RNA transcript. There are 2 DNA strands in this picture. One of them is actually forming hydrogen bonds with messenger RNA. That's the template strand. It's also called the antisense strand. Up top, we have a strand that will be entirely left alone, and it's going to be called the coding strand. Here's why. Let's say the coding strand says A, T, G. That means that its complementary DNA strand said T, A, C. And let's see exactly what that means for our messenger RNA. Our messenger RNA is complementary to the template antisense strand, so it will have A, U, G. What we can see here is that your messenger RNA transcript is basically the coding strand, except we have exchanged uracil for thymine. So in order to have transcription happen, we used the properties of DNA structure again. Because DNA is anti-parallel, RNA polymerase can only add on to the 3' end of messenger RNA. Also, we use the complementary base pairing rules in order to make sure messenger RNA is accurately carrying the information contained in your DNA. We also used complementary base pairing so that the transcription factor could find the ta-ta box. What happens next only happens in eukaryotic cells, and it's when we take this messenger RNA and we process it. So step 2, messenger RNA processing, eukaryotes only. What happens here is we add protective end caps. That sounds a lot like telomeres. On the 3' end, we have the poly A tail. On the 5' end, we have the 5' cap. After we add those protective ends, we are going to splice this messenger RNA until it contains only the genes we want to form into a protein. We cut introns out and we keep exons so that we can express them. This mature messenger RNA can leave the nucleus. The best part about this process is it gives just one DNA gene a lot of options for the protein it's going to make. If we take that gene's messenger RNA and splice it 2 different ways, we'll end up with 2 different proteins. That is super efficient and means that we can have way more proteins than we have genes. Step 3. We had a transcript, we processed it, and now we need to translate it from nucleic acid to protein. So let's get a chain of amino acids going. The first thing that happens is initiation, and that's when your messenger RNA strand binds to a ribosome that's made of rRNA. Next, transfer RNA helps the process of elongation, which is when amino acids are adding to a growing polypeptide, because every codon has a complementary anti-codon on the transfer RNA. Let's zoom in. This is the start codon AUG on our messenger RNA strand, and its complementary anti-codon on tRNA is UAC. The final step of this will be termination, and that will happen pretty far downstream when your messenger RNA reaches a stop codon. Then your ribosome will release that messenger RNA strand and also release the growing polypeptide. There is one thing that can get in the way of this process, and it is when your mature messenger RNA meets an enemy in the cytoplasm. This is the last type of RNA that we're interested, and it's siRNA, or siRNA. It stands for small interfering RNA, and legit what it does is just finds a messenger RNA transcript and totally wrecks it. It eliminates it from the cytoplasm so that no protein can be made from it. Your big takeaway here should be that DNA structure makes it an ideal molecule for information transfer. It's protected by the nucleus. However, it is easy to unzip, either to make more of itself or to make a transcript that is disposable and can travel throughout the cytoplasm to the ribosomes, where the ribosome can make it into a protein. There are a lot of different types of RNA, and they all use the base pairing rules to make accurate biological processes. So to review all of our RNA, we have messenger RNA that can leave the nucleus. We have ribosomal RNA that makes up ribosomes, transfer RNA that carries amino acids to their proper codons. And finally, we have small interfering RNA that can take a messenger RNA transcript and totally ruin its life. And that's all we got.